Welcome, everyone, please. Today, it is our extraordinary honor to welcome on behalf of the major issues in modern biology lecture series, and as well as on behalf of the plant biology graduate group, Professor Dr. Dirk Inze from the Ghent University in Belgium. Dr. Inze was so kind enough and obviously undeterred to extend his trip to the US from sunny Colorado to where he attended the Keystone Symposium to California as it is right now. But in fact, he brought us the first sunshine yesterday within weeks. Dirk Inze received his PhD in Jeff Shell's laboratory on mechanisms of agrobacterium-mediated plant cell proliferation. Shortly after that, he was appointed as the research director of the joint laboratory between the Ghent University and the French National Institute for Agricultural Research, also known as the INRA. It was then and there when Dr. Inze initiated his career-defining research programs on plant cell cycle control and plant cell death. Dr. Inze is currently not only a professor at the Ghent University, but he is also the scientific director of the Department of Plant Systems Biology at the Flanders Inter-University Institute for Biotechnology. And there he heads a little, scientific enterprise of almost 200 researchers and employees. And in addition to that, he is also a proven successful entrepreneur, because he has founded and that's what I found out yesterday, the thriving biotech company Crop Design. Dr. Inze is an elected member of the Royal Flemish Academy of Science and the Arts. And of course, he has received numerous prestigious awards for his groundbreaking research. He will tell us about today in his lecture on cell division and growth control in plants. Thank you very much, Dirk Inze, for visiting UC Davis.
Yep, thank you very much, Stefan, and thank you for attending this seminar, even so close to Easter. So what I'm going to do is try to stay away as much as possible from molecular biology and trying to talk a little bit on cell division and why and how actually our cells are divided and what will be the future of our research in the coming 5 years to go. Well, just anyhow, I have to introduce a little bit the cell cycle, and I think it should be known to you that this is a highly regulated process in which, in fact, which serves the purpose of replicating the genetic information and during the S phase, and then separating this genetic information over the two daughter cells by mitosis and by cytokinesis. And this process is just interrupted by two gap phases. Well, in the last 10 years, in fact, we learned an enormous amount of things about cell cycle regulation in plants. And what is so nice, in fact, that it turns out that this is a very evolutionarily conserved process from yeast to man, and many of these components you can find back in all these different organisms, but certainly the regulation of these components is very much species and specific. And because I need all this to explain some of the data, essentially cell division is driven in plants, like in any other eukaryotic organism, by these cycle-independent kinases. These are serine, threonine, protein kinases. And in fact, they only work when they associate with the cyclin. And this complex, in fact, will propel the cells to the different phases of the cell cycle. This is a very, as you might anticipate, a very highly regulated machinery, because this complex is regulated by transcription, by proteolysis, by phosphorylation, by interaction with inhibitors. So remember also this name. These are small proteins which can bind these complexes and inhibit these complexes. And as you can see here, we have many of these genes in Arabidopsis. So there are many possible permutations possible on that complex. Now, without going into detail, we have a reasonable understanding on how the machinery actually works, and not going through that slide. You can find it in many different reviews, but we know how this machinery in fact works to propel, to push cells from G1 to S to G2 to M and so on. And I could talk an entire seminar only on that, but not going to do that. And rather just would like to leave you with a message that although we understand very well what's happening in individual cells, or reasonable well, that we have a very poor understanding on how this cell division machinery actually integrates with development, and how the environment, in fact, is using this machinery to make new structures, or to have reduced growth in environmental stress conditions, and so on. So that's what I'm going to talk about, and give you in fact, 4 different shots, in fact, of ongoing research in our lab. One will have to do, in fact, with understanding how new organs are made, and I'm going to talk about all this data is actually non-published. I'm going to talk to you about how root pericycle cells are going to make a new root, and how the cell cycle machinery is activated. And I'm going to talk about 3 different aspects of cell cycle exit and the onset of differentiation. One is induced by DNA damage. A little story on endoreduplication and what we know about that in plants. And I'm going to talk about the recent story on environmental control of the cell cycle. So first on how do cells make a new organ. And so a couple of years ago we took on the challenging question, in fact, to try to understand why these cells here, these are expressing a cell cycle marker. And so you could see that these cells in the root, and not those ones, these cells alone, are activated for cell division, and are going to make a new lateral root, in fact. So why we became interested in that, because it's an in vivo model for cell cycle entry. Only a few pericycle cells were susceptible, and auxin is of course an auxin triggered effect. And what is more important, that it involves an asymmetric cell division. And so this is actually a quite challenging question, because these cells, these few cells, in fact, they are coming from this cell layer, the pericycle cell layer, and even furthermore, it's only the cells which are opposite of the protoxylem poles, in fact, so only here or here, that actually will be able to respond. And in fact, this has been described in the literature quite extensively, and so we have the whole process that was already by the Benfi lab, in fact. But what interests us is this very first event. And this first event, I mean, when you make longitudinal section, you can see that this is always coincide with an asymmetrical division. So, in fact, we have been recently confirmed on that. This is a confocal image of a root which has been transgenic for a plasma membrane marker, an LTI gene. And what you can actually see, and you have here, this is epidermis, cortex, endodermis, and you see here the pericycle. These are the long pericycle cells, and this is so. In fact. When you treat all these roots in order to make a lateral root, what you can observe is that there is first an asymmetrical division from actually both cells, and this is immediately followed within actually 5 hours, 45 minutes to be exact, by a second asymmetric division, in fact, here, and then yet another, one, here. And so this, and then only then, these are all anticlinal division, you will have the first periclinal division. So we are interested actually in this first 2 asymmetric divisions, and why we... No, actually very little, in fact, yes, yeah. And why we are interested in that, because very much in nature, one sees that cells are just proliferating, I mean, making the same the same cells. Then you get, this is always goes together with symmetric division, and most often this symmetric division are always just along the shortest distance between the two sides of the mother cell. So very few you will find this along the longitudinal axis. But in many cases, like particularly in plants, when we have the formation of a new organ, you get what is called a formative division. And that's always associated, like, in fact, with a true asymmetric division, where you have a different cell content here than like here, or where, in fact, the cell, even when they morphologically will divide symmetrically, that there will be still an asymmetric distribution of cellular compounds, like, in fact. So, and this is like a message which is true throughout, like in nature, like in fact in many different cases like of organogenesis. For example, the formation like of stomata, like in fact, but also in Drosophila, neuron development, C. elegans, and so on. Like in fact, embryogenesis, and in fact, the first zygote which is going to divide asymmetric to form the suspensor and so on. Like, I mean, there's all examples like of this kind of formative division and how it relates with the formation of new structures. Of course, it shouldn't surprise me. This is a technically challenging thing to do, like in fact, because this root like is something here at this stage, something like 5 millimeters like in size, like in fact, and we are focusing on these two cells like here, which are going to divide asymmetrically. In fact, you can see this here, and just opposite of this protoxylem poles. Fortunately, like we found that a trick to get more of these cells. And the trick is actually very simple. It's you actually have to treat a root with an auxin transport inhibitor, NPA. This auxin transport inhibitor, in fact, will repress the formation of any lateral root, but would you keep cell cycle activity in the root and shoot apical meristem, as visualized by the cell division marker here, the cyclin B. But what is the interesting thing of that, if you add auxins to such treated roots, you can see that the entire pericycle is now labeling blue. And this is a labeling blue opposite of the protoxylem poles, like in fact, and the entire pericycle will, after days, like in fact, will start to make lateral roots. So what this MPA is doing is, in fact, is priming like all these pericycle cells so that you make the system just synchronously, or more or less synchronously, is going to react to auxin, and it will actually, you fool the system. The pericycle will start to initiate, like in fact, this whole lateral root asymmetric division process, like in fact. So this has been very well characterized, and since it's all been published, I will not discuss all it, but in short, what is happening here in hours after that you have put this NPA-treated seedlings on auxin, you can see that you have, first, like within 1 hour, you see the auxin responses, DR5, and after 3 hours, the entire pericycle will have stained for this marker. And a little bit later, like in fact, like you start to see the expression of a G2 marker. So this is, I must say that all these root pericycle cells are in G1. So what auxin is doing is really setting a program that the cells will move synchronously through S and through the G2 phase of the cell cycle. So because the entire root pericycle in fact is reacting, we are already in a better situation. We have actually much more cells in fact already, not these two cells, but we already the whole pericycle and just opposite of the protoxylem poles. So we decided actually to introduce another sophistication in the system by now sorting this pericycle cells. So in fact, we've been sending a student over to Phil Benfield lab to learn all the technology, and now we have it run it on our own fax in our department. And by using actually a marker, this from the Jim Esselhoff collection, and this marker is only expressed in the pericycle, actually not in the root meristem, only in the pericycle, and in fact it's opposite of the protoxylem poles. So by using this particular system, and by using this inducible system, so we're using now the Jim Esselhoff marker line, but we're doing exactly the same. We treat this with MPA and we add auxin. So, and by using different time points, so by using, of course, a 0 time point, arrested pericycle cells, we used like a 2 hours time point. And when this has been shown to be involving auxin, you will see that later. And cells are still at just or going to enter the cell cycle at the G1S transition. And then a second time point 6 hours, in fact, which already cells are already starting to express this G2 marker. So they are actually all in the G2 phase of the cell cycle. So, and this would allow us, in fact, to identify components which are involved in the asymmetric cell division. And I will show you that's actually also the case. So this experiment was set up. It's root apical meristems were dissected away. So we only we only took segments of the root itself at large scale into 2 independent experiments, and data were then analyzed on those Affymetric cell chips. And to make a long story short, in fact, 920 genes, in fact, are differentially regulated and in this system. But you can cluster these genes in different groups, and I will discuss some of these genes. Well, first of all, what the bottom line, but I'm going to discuss a little bit more, that it is possible to find genes involved in this asymmetric cell division. And already here, I will give you the names of these genes later. For example, in this cluster here, this is 0 hours, 2 hours, and 6 hours. This is a gene cluster of genes which are specifically induced after 6 hours after this auxin treatment. Well, you can see here, this one is actually only expressed in these 2 asymmetric, the smallest of these asymmetric cells. So that this is after that both the 2 long pericycle cells both have divided once asymmetrically, and this marker is only expressed as asymmetric cells. This one as well. Some genes are repressed, and you can see this here. This is all clearly, in fact, but I come back to that in a minute. In fact, the whole system allowed us to identify a whole bunch of very interesting new genes involved in this very early very early root development. And what you can see here is that when you knock out, this is just a selection of genes that I show you. When you knock out some of these genes, in fact, you also have actually reduced lateral root formation. That is as far as we are. So what are these genes now? Well, it turns out that if you look to this lateral root initiation process, and this is also from earlier work which we published, the very first event that we are seeing is the onset of a whole auxin signaling cascade, followed by the onset of the cell cycle machinery, first G1S, and then followed in fact by this asymmetric cell division. So let's maybe first take a look into that. So if you look to all the auxin regulated genes on this chip, in fact, you can see that there is a whole class of genes which are falling in these this clusters. So some of these genes, like in cluster 5 and cluster 10, are temporarily induced after 2 hours. So here, or reduced, some in this cluster 4 or reduced. You can see that there is, and that is apparently also talking to John today, in fact, and there is really a totally reprogramming of this auxin signaling cascades, what are going on in a very, very short period of time at the transcription level. So going a little bit further, in fact, now cell cycle, here, if you just look for the cell cycle genes, you can, these are all different cyclins, and so you can see that this, that almost all these genes, in fact, cluster in this group tree. So these genes, in fact, which are very low expressed after 2 hours, but when the whole cell cycle machinery is setting off, they are very highly expressed. And then maybe the most interesting, I mean, we also actually functionally validated many of these cell cycle genes, and what is shown here is that this, for example, the cyclin Bs and so on, that they actually indeed are expressed in these very young, very early founder cells for these lateral roots. So then we go to this, what we expect, because there's an asymmetric cell division happening in that system, that we're also going to find a set of genes which has something to do with this asymmetric division. And that is actually the case. So when you use gene ontology and you ask the question, which cluster do I see genes, in fact, which have as some homology, but what is known by the gene ontology as asymmetric division. There are a number of genes involved in asymmetric division, described in Drosophila and so on. So we're looking actually for homologs in plants of those genes. And what you can see here, that there is an enrichment of that annotation in cluster 3, also in terms of polarity. That's another buzzword you can zoom on, in terms of cell fate here, and on G to M transition. So this cluster 3 here, this one, so genes which are upregulated amongst around 6 hours after this auxin treatment, is very much enriched for these terms and for genes which have something to do or have homology with genes known in other systems with asymmetric division and polarity. So we actually focused on this cluster, and in fact, for this 920 genes, in fact, if you take this as a criterion, in fact, then this cluster 3 contains 340 genes upregulated in the asymmetric division during lateral root formation. Now, so in order to narrow that further down, in fact, what we have been doing, we have been looking, because we disclustered not only as genes which we think has something to do with this asymmetric division, but also genes that have shown which have something to do with mitosis in general. So what we have been doing is to actually subtract this 340 genes by genes which are expressed during mitosis in dividing cell cultures. In actually using the data from Menges and Murray, in fact, in what they have been doing, they have been looking to cell suspension of Arabidopsis, and they have been looking for genes which are expressed in S phase, G2 phase, and so on. So we have been subtracting these set of genes from that cluster, leaving something like 190 genes which are potential in cell fate and cell polarity, which is still a very large number of genes to deal with. Now, we are confident, and I'm going to only to show 2 of these genes, we are confident that amongst this set, there's a strong enrichment for genes involved in asymmetric division. So one gene which we're actually characterizing further is an homolog of the crinkly 4 homolog. Actually, it's a receptor-like protein and a kinase. And so what this gene is actually doing is the following. You made actually now recently GFP fusion to hit and under control of his own promoter. And so we are comparing here the picture I showed you before. This is this pericycle cell layer. And now we are comparing the same stages, in fact, the same timing with transgenics, with expressing the promoter crinkle homolog, in fact, and what you can see here is that when the cells are dividing asymmetrically, you see an asymmetric actually expression of these genes, in fact, and even of the stage further, you can see it here. Turns out to be an interesting gene, which is only as expressed, in fact, in the asymmetric, the smallest of the 2 cells which are after asymmetric cell division. So another one which I showed already before, which solves exactly the same pattern, is actually an expansin, rather surprising because you would not think that. So here again, it's now a promoter Gus fusion, in fact. And you can see that after that, only in the 2 smaller cells after this asymmetrical cell division, that this particular marker is expressed. So it's in fact is quite exciting for us, but we are now in the process of further working out what these genes are doing, functional analysis, and so on. So this is what I wanted to tell you about ongoing work to understand the asymmetrical cell division. So the next thing I would like to talk a little bit about is on cell cycle exit. We have a very poor understanding of how cells are exiting the cell cycle. Remarkable, and this of course is very important because at some point in development, cells have to stop to divide and then start differentiation and the process. So, because actually here in the UC Davis, the group of Anna Britt works on DNA damage. I thought it might be interesting to show also a little bit on what we are doing on DNA damage and checkpoints. So just to make and to generalize the story, there's a pretty good understanding, in fact, on how cells sense, and this is taking data from the mammalian field, DNA stress, in fact. And one should distinguish pathways which, in fact, detect single-stranded DNA breaks, like, for example, a block in the replication fork, or interfering with DNA replication as such, like hydroxyurea will deplete the cells of nucleotides, and thereby, in fact, the replication will stop. Acetylcholine is a drug from a fungus which interferes with polymerase activity. So any block in DNA replication will activate a kinase, which is ATR kinase. And this is called the ATR pathway. And then double-stranded DNA breaks, that's like gamma radiation and zeosine, it's a drug which induces double-stranded DNA breaks, will in fact induce another kinase, in fact this ATM kinase. And both of these kinases will induce the genes involved in DNA repair, but what more importantly, they will also induce the two other kinases, the so-called check kinases, check 1 and check 2. And what interests us, in fact, is how they affect cell cycle progression. And well, I can tell you, the way that this works, I think, is through a particular kinase, which is called the V kinase. And what this V kinase is actually is doing, it's a kinase which is phosphorylating these cyclin-dependent kinases. You remember, maybe I should have introduced a slide here, cyclin-dependent kinases are central to the progression, in fact, of the cell cycle machinery. Well, this V kinase is putting a phosphor group on tyrosine 15 on these kinases and thereby inhibit this kinase activity. So it's a kind of, it arrests, in fact, cell cycle progression. I mean, at least that's what the way it works, in yeast. So we actually recently cloned this kinase, and what you can see here, and this has been also shown for a number of other stress conditions. So in other conditions which causes DNA breaks, it rapidly induced by all kind of treatments which causes DNA damage, like here, zeosine inducing double-stranded DNA breaks. And this goes, in fact, with more or less the same kinetics or the same dose concentration on typical markers like this PARP here, this poly-ADP ribose polymerase. This is a marker for repair, in fact, for DNA damage. So we isolated, this is, again, this is in an ATM, and we know also in an ATR-dependent way. And so that's, if you would, you cannot, this now by gamma irradiation is another example. So normally, in fact, in the wild type, the V expression is induced by gamma irradiation, but it is not expressed in the ATM mutant. So we have now recently a knockout in this V kinase, and to our surprise, these plants are morphologically completely normal, and we actually measured all kind of parameters, leaf area, number of cells, and so on. It's indistinguishable, also in terms of endoreduplication. I come back to that. And however, these plants are hypersensitive to DNA damaging drugs. So in fact, here you can see that when you germinate these seedlings without hydroxyurea, this indistinguishable. However, when you treat all this, or when you germinate the seedlings on medium with hydroxyurea, which is in fact causing, in fact, a DNA replication stress, you can see that there's a reduction in the wild type, but and this is phenotypes look very much like the ATR, ATM mutants, that in the absence of hydroxyurea, the V1 knockout, and in fact, they're at least the primary root, doesn't seem to grow much further. And similar results were obtained with acetylcholine, but also with zeosine. So it's not only single-stranded DNA breaks, but also double-stranded DNA breaks. In fact, we know that this is due to a cell cycle arrest because we made an inducible construct in which you can overexpress this kinase, because constitutive overexpression is lethal, and this is in fact consumes almost the entire meristem. The cells are not in very great shape, here, so what we think is happening here, that DNA damage in plants, in fact, will through the ATR-ATM pathway induce the V expression, and that inhibits, in fact, cell cycle progression. At the same time, in defense systems, like I showed you for the PARP, will be induced. When you have a V knockout, what will happen is that in condition of DNA damage, of replication test, in fact, the cells will not be able to censor that, will not be able to turn off the cell division, and thereby, in fact, will just actually, you get the kind of lethal phenotype. And this actually, this continuation of cell division in the presence of DNA damage might actually cause cell death, because, and this is a recent experiment, if you treat, so if you treat roots, and treat in this case with afidicoline, it's the same with hydroxyurea. If you treat, so this root cell here, this one, if you look what's happening here and you're going to stain them with Evans blue, which labels the cells undergoing cell death, and in fact you can see that they're blue, they stain much more blue, indicating that there is actually active cell death going on, maybe because you keep the cell cycle machinery going, and in fact in the presence of DNA damage might trigger a programmed cell death mechanism, what we believe. Okay, so unfortunately we thought that that also many different environmental stress conditions, the V kinase might play a role in sensing DNA damage and turning of the cell cycle, but that does not appear to be the case. Like the V knockout is not hypersensitive to Sol's test, for example. So there must be other mechanisms which interfere with cell division during environmental conditions than this one. Okay. Now, a little story on endoreduplication, and this is an intriguing phenomenon. Intriguing phenomena is that in many plants, and you find that the cells in the normal cell cycle is the cells are in G1, they replicate their DNA, so they come in G2, so have a doubling of their DNA content, and then they divide the DNA over the two daughter cells. It appears now that actually in many, many different plants, in fact, this distribution of the genetic information of the two daughter cells does not occur. The cell will, after one round of DNA replication, will have another round of DNA replication, yet another, and so on. So you get actually polyploid, in this many copies of the DNA in the same cell. And you can see this here. So each time you have the G1 cells has a 2C DNA content, C being the size of haploid genome. After DNA replication 4C, that's a G2. But then 8C, 16C, 32C, that's just amplifying the DNA. And what you can see like here is that in older leaves, like of Arabidopsis, for example, you have even 32C values. It's only in these cells which are undergoing or going to make the gametes, you find only 2C and 4C. So it's actually not very well known what the reason for that is. And I will discuss some of the physiological consequences of endoreduplication and trying to give you some views on that. What I can tell you, and I'm going to be very brief on that we have now, and this has all been published. I'm not going to bother you because it's rather complicated, but we have a fairly good understanding on what controls endoreduplication. Why do you find it in older leaves and not in the younger leaves and things like that? And this is actually a cell cycle, and so this is now the current view on what regulates, in fact, the G2M transition. So it turns out that plants which undergo endoreduplication, that any block in the G2M transition will actually, as a default, will make that the cells, in fact, will start, at least some cells in that organism will start to back the DNA replication. So you have a shortcut of a cycle. So we have identified over the years now many genes, and not only us, but also the lab of Condorosci and Crisanto Gutierrez. We have identified many genes, in fact, which have something to do with endoreduplication. And as one of the master control genes, at least the furthest down in the pathway, is this DEL1 protein. And that's, for example, one of the proteins you can use actually to manipulate endoreduplication. What is this a kind? This is actually a transcription factor. Actually, it's a transcriptional repressor. It belongs to a class of proteins, the E2F-like proteins. These are actually normally regulators of DNA replication. And this is a special class effect, which was first discovered into plants, and only later, actually recently, human homologs were identified, E2F7, E2F8, and they have turned out to be important regulators of cancer. So it was a good example that plants can lead the way to cancer research. Anyhow, these proteins, which are called DPE2F-like DELs, in fact, they can bind to promoters, in fact, of genes which have something to do in DNA replication, but they act like as a repressor. So we have shown, and this was recently published in Current Biology, that this DEL turns out to be master regulator of endoreduplication. So if you overexpress this DEL1 factor, plants looking pretty much normal, maybe a little bit smaller. Similarly, when you have a DEL knockout, however, when you start to measure levels of endoreduplication, and this is not, you can see like here that DEL overexpression reduces the level of endoreduplication. This is now looking to just a leaf at a given developmental time. You normally have the 8C peak, this is undoubtedly this is endoreduplication, 16C peak, so you can certainly see a reduction like here in the overexpression line, and just the opposite, they have a knockout, you have even a 32C value, so it means an extra round of endoreduplication. Now, and we know well that this particular protein is regulated by itself during leaf development, in fact, exactly this DEL1 protein levels disappear, and this goes then together with induction of yet another one. And so we know many of this data, and I will not bother you about it. Just remember that DEL overexpression, in fact, is reducing the level of endoreplication, and DEL knockout is in making effect that you have more endoreduplication. So what is now the role of this endoreduplication? Well, one of these ideas you find in the literature is that it supports cell growth. And so there are many data which seem to indicate that this is actually true. I'll show you one, this may be not the best picture, but like in trichome development, there are many mutants known which affects the number of branches in trichomes. And this correlates with level of endoreduplication. So mutants which have only 2 branches, they have a reduced level of endoreduplication, 16C. The normal 3 or 4 branches have 32C, and then you have ones with 64C. So the larger the cell is, the more of DNA that you have in this cell. These are single cells, and the nucleus has to undergo different rounds of endoreduplication. So you might, and there's indeed a correlation, in fact, between cell size and the DNA content that you can see here. And you can do experiments simple like that. This is actually taken from a paper from Jan Traas. This is just it's a shoot apical meristem, and this is treated with a drug, orizaline, which blocks interferes with microtubules. So this blocks actually the progression through mitosis. And what you see, in fact, is quite remarkable when you prevent cell division and just keeping the cells in mitosis, that cells actually grow. You can see that here. Right? So there's no division anymore. The meristems, and even we will get the formation of new primordia. This is only when you block the cells in the mitosis, not when you block them in the S phase by, for example, hydroxyurea. That doesn't work. And And in a nice work, he showed, like, in fact, that there is a direct linear correlation between the DNA content, so this growing, and in the area of the cell. So in order to grow, so you still need to amplify your DNA. So that's an... and also in our transgenic cell, in fact, this is, so if you look for a leaf, and you're going now with microcytometry, you're going to have to measure the DNA content in individual leaf epidermal cells. You find a kind of mosaic. Often cells surrounding the stomata are 2C, and then you have 4Cs, like an 8C and 16C cells. And you can see that there is a direct correlation between cell sizes and the C value in these plants. And if you disturb that, like for example in a del overexpression, remember del overexpression, less endoreduplication, well, you just see that the ploidy, like in fact, I mean the cells are also, the average cell size becomes much smaller. So this is direct correlation, and certainly, I mean, one can conclude from that the cell volume positively correlate with the level of endoreduplication. So this is an important concept. Because we always think that growth is in fact is cell division, so you have cell numbers, like in fact, and in addition to cell division, you have to have cell enlargement. And these are the two components like of growth, because cell division will just half the volume, so you have to add cell. So, and we always think about cell cycle as being only controlling the cell number. And I think it's a wrong concept, because at least in some plants, like in fact, cell cycle might have something to do directly with cell enlargement. So that is certainly, this is strong correlation, and it's another way of looking to the importance of cell enlargement. However, the situation is not so clear, so there's a strict correlation within one organism between cell size and the endoreduplication. If you look a little bit across borders, like in fact, so here we're looking in fact like to whether there's a correlation between the level of endoreduplication and the mature cortical cell length in the root now, in between something like 20 different ecotypes in Arabidopsis. And you can see there is absolutely no correlation whatsoever. There is, so for example, Wasilewskaya as 20% of the cells in 8C and 16C. So here you cannot discuss about. This is certainly endoreduplication. And you can see like here that you have a very large cells, and while CVI, for example, has more endoreduplication, they have the smaller cells. So the correlation does not really hold between different genotypes. It only holds within the same ecotype. So another role for endoreduplication has been set is to support metabolism. And this, for example, in endosperm, and we eat every day, like in corn, like in rice, endosperm is highly polyploid. I mean, sometimes you find 64, like under 28 C values without any, even more, like in fact. So this, and it's been settled that this is support on metabolism, so that you have more copies of DNA to support protein synthesis or oil synthesis and those things like that. Now, I can tell you that the experiments of Brian Larkins here in the US has shown now recently that there appears no correlation whatsoever between the level of endoreduplication and the capacity of, in this case, corn in endosperm to make, for example, storage proteins. So by using, in fact, transgene technology and by interfering with this cycle-independent kinases, they were able to make plants which have significantly less, in fact, or actually several rounds less of endoreduplication in the endosperm, and these guys, they make the same amount of zein. So that doesn't seem to be correct as well. So I'm going to show you another piece of data that endoreduplication might have something to do with environmental stress, and why is that so a tempting idea? And if you look in many different species to the level of endoreduplication, then there's a remarkable correlation between the presence of endoreduplication and the lifetime of these species. So short-lived species, so species particularly which grow like in the beginning of the season or in the end of the season, so in the spring, like of autumn time, they have very often a lot of endoreduplication. While species like wooden species, which grows like for hundreds of years sometimes, there is virtually no endoreduplication. So I mean, there's no correlation whatsoever between size of an organism and whatever. So it's really to do something with the ability to do something very fast. And so don't look this as points. Actually, the points are different tissue. It doesn't really matter. Like in fact, but there's a strong correlation. And so we actually we set out to test on that whether maybe just having more DNA copies would buffer against inactivation by mutation. So the idea was, maybe when the species, when they're short-lived, and they often grow in the early season or late in the season, like in fact, and just to have that maybe they are exposed by too much hard shell factors, maybe having more DNA would just buffer it. And this turned out to be quite an interesting correlation. This is now an experiment. It's rather premature, but it's reproducible. That is, for example, if you manipulated the level of endoreduplication by this del, you remember del overexpression less endocycles and del overexpression more endocycles. You can see, I hope you can see it here, that when you have less endocycles, the cells are more sensitive to UVB light, and when you have more endocycles, they're getting more resistant to UVB light. So, which may be just more DNAs, maybe less chance to get mutations, and you have many copies of the same alleles all the time present. However, I have to say that might not be the only part of the story, because it turns out when we did actually a microarray on these del plants, and both overexpression and the del knockouts, and there is something which bothers me, that one of the top regulated genes, so upregulated in the del knockouts and down-regulated in the del overexpressions, in fact, is this type 2 CPD photolyase. And this is so it might be that this, besides regulating endoreduplication, maybe that del factor, so that might also regulate this photolyase, and this photolyase is actually involved in DNA repair. When you make a knockout in that particular gene, actually they become sensitive to UVB light. So it's still too early to conclude whether it is only endoreduplication or that by accident this factor also regulates something in UVB repair. There might be actually another reason why in short-lived species, in fact, you have a lot of endoreduplication. And that maybe is a very simple reason. Actually, you don't find this in the literature, but maybe it's just about economics. If you think about, if you have a space, you want as fast as possible making the leaf. You have little time because to make some destruction, because all the other plants are starting to grow as well. So, I mean, if you have to make a leaf, either your strategy would be you make many cells and you put every cell a nucleus and they make it all very nice and very. But why just having filling the same space with just having one big nucleus supporting larger volumes, and maybe just like that. And that's something with this mutant. Actually, we can now in a more ecophysiological experiment we can test. So. Do I still have time? But I have a story of something which takes 5 minutes, which brings me 2 minutes over time, I think. I want to say something short on an environmental stress, which I think environmental stress, I mean, we all know reduces plant growth, and I will not talk too much about it. We have a very pure understanding of why this actually happens, and sometimes you find also that there is a modified architecture. So many plants escape stress because you get much more branching. In fact, you get reduced apical dominance. And we got recently involved in that because an interesting connection we found in another project, a gene which was induced by oxidative stress. It's called prohibitin, and it's actually not going to talk about details. You can see here, as soon as these plants are exposed to hydrogen peroxide, they start to transcribe rapidly this prohibitin. And it took our attention because it's an E2F-RB interactor in mammals, and it's also a molecular chaperone of respiration chain. So it's actually a dual function in animals. And this E2F pathway, maybe many of you will know that, but this is It's actually a system that to regulate the expression of G1S gene. So, in fact, this is an heterodimeric transcription factor which binds to promoters of DNA replication genes, like in G1 is kept inactive hyperphosphorylation by the CDK cyclin. So that's a mechanism by which these things work. Will hyperphosphorylate it and thereby activating the transcription of this DNA replication genes. And so we thought maybe prohibitin is induced, like in fact, and it was shown like in animal system that prohibitin represses cell cycle progression. So maybe that's the way that you have reduced the growth, because you actually, so what it was doing is represses E2F responsive genes, by recruiting represses histone deacetylase in particular. And, but there's in animal systems is a second function to describe to it, which is a function as a mitochondrial chaperone. And what they are doing that, like in fact, they are protecting the respiratory chain, like in fact. And if you have a disruption of that gene, it causes lethality and accelerated aging and so on. So it's an essential gene with a dual function in animal systems. Well, it turns out that in plants, in Arabidopsis, we have 7 of these members, 5 are true genes, the 2 others we have not been able to clone seed in it, so we are in the, I think it's maybe pseudo genes. And but what is interesting that in plants, these proteins are only mitochondrial, so apparently the nuclear function in E2F is not being kept. So by fusion and a number of other experiments, like if you fuse one of them to GFP, and co-localize with the mitotracker, it's mitochondrial. So we have evolutionarily, we have that these proteins are just doing actually the mitochondrial function. So you might ask the question, what is the relation now with stress? Well, first of all, this was very nice to see that these genes are expressed in dividing cells, and any dividing cell, also dividing stomata, and also in fact they are induced by auxins, also in this pericycle layer, but what is most interesting here that we found that is when you overexpress these genes, that you get a clear phenotype, and this phenotype is just increased branching, less apical dominance, in fact, and now the connection with stress comes, in fact, if we use the same plants for microarray analysis at large scale, in fact, and many genes are upregulated, and we'll not talk you through that, but just tell you when you look to these genes, in fact, and you do a meta-analysis, so it means comparing that data set with many other data sets, in fact, we found that this regulon, that is the regulon which is induced by this prohibitin overexpression, is in fact also a regulon which is very strongly upregulated during abiotic stress conditions, salt, UV light, oxidative stress, and so on. So you can see that here. And this was further confirmed by looking to a large, more targeted data set. So looking for co-regulation, in fact, and it turns out that in the prohibitin data set, there were at least 5 genes, in fact, which were always strongly upregulated also when you have environmental stress conditions. So prohibitin, just overexpression of that protein, induces very specifically that regulon that is also happens to be overregulated, overexpressed in stress condition. So why is that? In fact, so in order to figure that out, this UDP glucosyl transferase caught our attention. And to make a long story short, in fact, this is a member of a very large gene family, and what it is doing, it's placing a glucose residue from UDP glucose on a substrate. And that substrate can be, it's been shown, I mean, it's known for a few of them, it can be IAA, salicylic acid, lignin precursor, abscisic acid, and so on. And maybe we thought maybe that maybe this gene here is also converting a substrate into something else, and maybe that is the cause why you have branching. And that turns out to be correct. So we have actually overproduced this enzyme, and just tested with a number of hormone combination, UDP glucose, and so on. And to make a long story short, we found that this indole-3 butyric acid was the substrate of this particular enzyme. So which is, all of you know, of course, IA, auxins, but there are also other naturally occurring auxins, and one of them is indole 3-butyric acid, in fact, and that is occurring a little bit lower amounts. Very little is known about it. Everybody's fishing in the same pond, in fact, we know that this is interconvertible, but we don't know if ABA is regulating something by itself. It's a true plant hormone, maybe a little bit different from auxin, or it just is a storage form for auxin. In any case, what this enzyme is doing is converting ABI into ABI glucose in the storage form. And it does not do the same thing, it doesn't do anything at all with IAA. So what we believe, in fact, we believe that this enzyme is responsible for the branching phenotype, because when you overexpress this, you see a 100% correlation between the expression levels and the branching phenotype. So what this is, since it's converting an auxin, IBA, indole 3-butyric acid, into a storage form, I mean it would reduce the auxin pool, reduce the apical dominance, in fact, and thereby more branching. So what we believe, in fact, is last slide before the acknowledgement, is that what stress is actually doing is, in one way, stress, we believe, is sensed by the mitochondrial respiration chain. And which makes sense, in fact, when the conditions are not so good, then, in fact, mitochondrial respiration the chain will be disturbed, in fact, this will be sensed by the prohibitins, and as a result, in fact, a regulon will be induced, which makes that this, amongst others, that this glucosyl transferase is induced, converting auxin into an inactive form, at least ABA, causing reduced apical dominance. So maybe it's a way that plants sense, in fact, environmental stress and convert this signal to an altered growth, altered growth habitat. And so this is, I think, quite a novel, interesting aspect in what's going on. So just to acknowledge some people, and so I presented work of Christophe de Sutter and Sergei Kushner with Lieven de Veilder on the V kinase, Yves de Smet, Stefan van Este, and Tom Beekman on the induction of the root asymmetric cell division, Veronique Baudolf and Aurine Verkest on the endoreduplication, and Olivier van Aken, Brigitte van der Kotte, Chris Moreel, Wout Bourjan, and Frank van Breuzegem on the prohibitin story. And this is done in collaboration with Hillel from the Tel Aviv University, and with the help for the microarrays and the sorting initially from the Phil Benfie lab. Thank you very much, and excuse me for speaking a little bit longer. Yeah.
Thank you very much, Dirk. Open to questions. Yes, please.
I was curious about your polarity markers. How many you said there are 300 something?
Well, we have now, the set is down to 190, and many are unknowns into that. So, what I can only show you at this moment, that we have confirmed, at least for a number of them, like this crinkle, crinkly homologue and the expansin, that they really are only expressed in the smallest of the two cells after asymmetric division.
Your initial criteria for choosing those is because their expression.
Yes, that's correct. I think because many of just have an unknown annotation, so it's rather difficult to tell something about that. Yeah.
Any idea why expansins involved?
No, it's a very good. Well, you would actually expect the opposite, isn't it? That it would be a million the larger cell, but that's not the case. I have absolutely no idea. No, it was one of the surprises. It's going to be interesting biology, I think. Who knows? I cannot answer it. Sorry.
Ben, please.
I just wondered if you looked for this pathway in different architectural types that are found, say, in the family.
So the last story on the prohibited, you mean? No, we have not done that yet. But that's, of course, it might be it was totally unexpected for us. Like, and what is the most surprising that it actually only deals with indole butyric acid. And I mean, if you look in, I mean, there's a lot of old literature, even not in PubMed, like on that. Like, but I mean, nobody. I mean, as I said, everybody's fishing in the same pond, so like, and then nobody looks. And then one of the intriguing questions whether there is any specificity, like in maybe IBA might be have slightly different effects on than IE. I don't know. Like that's why only IBA and not IE, and then what kind of variation do you see in nature? Like maybe it explains some of the bushy phenotypes, and again.
Did you have a chance to look at the UGT74E2 knockout and that intracellular?
Well, it's the problem. There's a problem. There's a closely related gene to that, and they are actually linked, and so it's difficult to get a double knockout. We do not see a phenotype with a single knockout, and so we have to now using an RNAi approach, but we have not the data yet.
Is there? You were talking about the relationship between endoreduplication and cell size, and I was wondering if there was any evidence that environmental factors that regulate cell size also might regulate endoreduplication. So such as light regulating hypothetical cell size.
Yeah, we know that for cell growth you actually do not need endoreduplication. So you can make large cells without endoreduplication. You can actually demonstrate all that genetically as well. But that doesn't mean it's like the fact that you have endoreduplication is a kind of facilitator. I think it's easier to make a larger cell so it's the way I see it for the moment. But there's no strict relationship between the two. So I think it's all about, honestly, I think it's all about economics. In fact, it makes really sense, the occurrence of endoreduplication in this short-lived species. And from an economic point of view, it might be just much more easier to deal with generating fast spaces by having less cell walls and all this. That's what I think. But it would be very interesting to test this more from an ecophysiological point of view. Maybe having the behavior of these plants in the kind of wild community. We have no mutants. I didn't tell you, if you combine some of the double mutants in this DEL with other genes, we can have plants which have virtually no endoreduplication, and so that even go further. And have plants which have much more endoreduplication, so they actually relatively physiologically normal in a lab condition, but maybe in a field condition they might behave to very, very different.
How are levels of endoreduplication correlated with the strength of gene expression?
Well, from the data from Brian Larkin's, what I showed you on the Zein, this is not our work, but the answer is no. And what is a surprise? But we actually are doing that. We're now sorting cells with different C values and comparing that. In yeast, there's been extensive comparison being done on diploid and tetraploid and hexaploid strains, and then you see that there is actually an effect which is not stoichiometric. So that most genes are just higher expressed in general, but there are also some genes which are over or underexpressed in the tetraploid. So it's not just a doubling, but there might be also regulatory mechanism. And of course, quite interesting, because if you think about it, many of our crops or polyploid, tetraploid, hexaploid, wheat, and so on. So there must be really interesting things going on there. And maybe that's also a mechanism to grow faster in a short period of time, what the breeders have selected for, and maybe it's also correlates to that. It's a little bit like finishing a life cycle in a short period of time, but it's just speculation.
Are there any more questions? If not, let's thank Dr. Inze again.
Thank you for coming. Yeah. Hi, Stefan.